Okay, the next presentation will be by Jayne Brim Box from the Northern Territory Department of Land Resource Management. She'll be talking on the impact of feral camels on rangelands, wetlands and vegetation.
Thanks, Steve.
There you go.
The people that are listed here are my coworkers, and most of them are in the audience, so if there are questions later, feel free to talk to them. We have a big collaborative effort across a lot of tenures and stakeholders, and that's been one of the pluses of the project, like people have talked about earlier today. 8 years ago, it was Glenn Edwards that said, Jayne, you go down to Uluru and figure out what those camels are doing to wetlands down there, because the TOs are saying they're running amok. So that was my start of working on camels 8 years ago. And the Northern Territory government tasked us with saying, figure out what damage is being done, assess the damage to water holes and vegetation. But within that context, really what we're interested in knowing is what's going to happen as these densities are reduced. This is our study area. We work in the southern NT. These other guys, Mark's talked about, there's a Peterman's, a Simpson. It's a large area. There's 500 kilometers between some of our sites. And just for comparative purposes, this is the ACT right here that looks a bit like a jalapeno pepper. Both of our asset areas are on Aboriginal land, and we do a lot of training with Aboriginal people, and they're an active partner in our project. And let's not fool anyone. I'm not born in Australia, so when I go to these areas with these land managers, I learn as much or more from them than they probably learn from me. But very active partners. Oh, I can see it here. I'm going to talk about the veg impacts first, and I always put a water slide in here because I'm an aquatic ecologist. So again, we needed a really big research team to get around this issue on vegetation impacts. And first, we've talked about this quite a bit today, and we're talking over and over about this idea of density and damage. And I'm interested in that too, because that's the crux of what we're trying to do in the environment. And so these are the three questions we asked. And Glenn had that graph this morning of infrastructure damage. And one of the, for us, the holy grail at the end of this would be, would we be able to establish some density damage relationship for vegetation? And Catherine Nano, my colleague that's done a lot of work on this, and she just has told me over and over, Australia is very complex where we live, inland Australia, and all these things are happening all the time. We don't avoid those. And is there a process going on where it's death by 1,000 cuts, not just camels browsing and causing these problems? Andy wanted us at the beginning of this project to pull a manual off a shelf and follow that methodology. Well, there are no methodologies for camels in the wild. So we did a lit review just like a good graduate student would do. And we also had Don Waller. We called him, he's one of the world's expert on browsing impacts. He came over in 2012 from the University of Wisconsin. And really what we wanted from Don was a lot of input and also to give us the golden star, a star to say, yes, you're doing something that makes sense. One of the things that I don't want to get stuck on is densities, because worldwide with browsing impacts, people have gone to more saying, what is the problem in terms of damage? So these browsing indices look more at the damage caused than the density of the animals themselves. There are a lot of study sites in this Andy alluded to before. There are 120 sites across these asset areas that Mark described. We measured altogether with our colleagues in WA and SA over 4,000 plants, sometimes 3 times within the study. So it was quite an extensive effort. Chris Brock did the stuff in WA, and he's here today. And because we weren't quite sure about our methodology when we began, and originally we were tasked to look at carbon sequestration in trees, we measured a lot of attributes of each tree and also pellet counts to see if we could get a relationship with local densities in this project. So we don't have seasons in Central Australia, we have boom and bust. So in 2010, we had an above rainfall year, average rainfall, and then it dried out, and then it burned. So it was green when we started here. It dried out, it burned, Then we had 157 days without rain, a record. Then some of the sites burned again. And then we had the hottest summer on record. So that kind of noise when you're trying to do science, to be honest, is a bit of a challenge. So I'm going to talk real briefly about the findings for our Northern Territory sites. They were both in high density areas. We had control sites in the National Forest where there are no camels. And the first question we asked were camels killing trees? In the browsing literature, elephants can be quite destructive and kill 20% of trees they browse. So that was the first question was, are these camels killing trees? We found little evidence that camels kill trees outright. So on the left is an acacia bush in a control site, and on the right is a really hammered acacia bush on one of the asset areas, but it's still alive. And if they're not killing these trees, what are they doing? Catherine was astute enough to say, let's split our trees and shrubs out because they're really different. So we have these shrubs that when they burn or browse, they can resprout basally. And then we have these trees, like mulga and Acacia oswaldii, that they need to recruit from seeds. So it's a different game when they're burnt or browse really heavily. And at the beginning of the project in 2010, what we found was for these key species we looked at, that the browsing was quite heavy across species. That was suspected, but it wasn't known. There are a few shrubs that they really didn't eat. But again, Andy showed this earlier, that means that when they go through a population of trees, they're eating almost every tree. That's significant. And if they're not killing them again, what do? So there's two ways that camels hit trees. So for the taller trees, they lose their biomass from below. And people have already showed this browse line. That's very obvious when you're out in nature. And then we really struggled, like, do they really go down and eat the trees all the way to the bottom? And our data suggests they don't. And what happens, these data, I'm not going to go into the stats, but I'll tell you when these relationships are significant. We found significant canopy loss in some of the tree and shrub species. So in this case, here's a mulga. And there are a lot of ecological implications for losing that much biomass on a tree. And one of them, Catherine, actually is a co-author on a study that looked at these perch pounce functional group birds. And we think there may be implications for them because they rely on these old age mulga stands for foraging habitats, and we really don't know what the impacts will be. So there's some nuanced impacts when you lose that biomass that we're not really across yet. And then shrubs. Shrubs are browsed from above. And this is mulga. This mulga escaped, so it escaped that browse fire zone. And all these guys are stuck, and they keep being browsed from above. And so if you browse from above repeatedly, what you expected, after a while, you just get stuck. And if you're stuck, you're stunted. So what this means, these guys on the right are the same height as these guys, but they're older. They're just stumpy little guys. We found significant stunting in a lot of the shrub species, and that's not surprising because most of their biomass is in that fire browse zone. So in our 3-year study period, this is a NAFI fire scar. I like to call this slide, no place to hide. Well, everything burned pretty much in the southern NT in that 3-year period. And browsing and fire both are really significant things that go on, but as Catherine pointed out, it's complex and it's really poorly understood what those interactions are. So in 2010, we had these trees, they burned by 2012. But the question was, are camels going to go back and target these trees? Or are they going to avoid these fire areas? What are they going to do? Is there going to be a pattern after these fires occurred? So what we found was on the left are trees that burnt. So the browsing went down significantly in the second and third years in the trees that burnt. Well, that's not surprising because the trees lost their biomass or their leaves were scorched. The thing we didn't realize was in our populations that the trees that weren't burnt were heavily browsed in those next 2 years. So what that means, just for clarity, on the left is a shrub that was burnt. It resprouted, but the camels don't touch it, and we think it because it's below that browse line. And on the right are whitewoods. They were just hammered in terms of browsing. So the end result is it doesn't matter if you're burnt or heavily browsed, that's what you end up with. A huge loss of biomass. So what does this all mean? We started with densities out here in our high camel density acid areas. And by now, and what Mark gave a really, I thought, excellent talk on where we are with densities now. Excuse me. Andy showed this graph before, but when we started in 2010, this is what was happening. As the camels were moving around, they were eating a lot of trees. And those trees, when they ate them, they ate a lot of branches. So that was a huge loss of biomass. Now at these densities, we know this has happened because we used data from WA where the densities were lower at the start of this study, thanks to Chris Brock and others. And so when the camels browse at those densities, they hit a few trees, and of those trees they eat, they only eat a few branches. So there's a significantly less amount of biomass being taken out of the environment. So here's the crux of my talk with the veg. We look at these legacy effects. So the densities can go up and down, but at these densities, what we started with, whether it's 2 camels per kilometer or 1, there are legacy effects out there. So there's too many camels because stunting and a significant loss of biomass has occurred. Down here at these densities on this end, it's not so clear. From the data we have from WA, it doesn't suggest there's a legacy effect yet. So the management, what's of concern now is where do we slide on this continuum in terms of this process? And now I'll switch to something I know about, and that's water. And again, we had really big teams we worked with. We had big areas that we worked in. This is a picture Jenny Davis took just recently. And I know you want to know this, so I'm going to tell you. We have lots of amazing water places in Central Australia, so there is water out there. And like others have said this morning, they can be quite significant sites. And some of our colleagues overseas, Don Seda coined this term. These areas are so significant for local people. They provide all these biodiversity outcomes, and also they're just precious jewels of the desert, and I quite like that saying. I was really fortunate when I moved to Central Australia 10 years ago. I had a mentor, and her name is Veronica Dobson, and she's a senior Ananda woman. And she was the one who said, Jane, these water holes need help. Can you help us? And she said they're being wrecked and ruined by feral animals. And what does wrecked and ruined mean? We saw pictures already today. It means the same thing in my study sites. And in this case, the things that are wrecking and ruining in many cases, these cultural sites, are feral camels. These are our water areas. I think Andy said before, we've sampled from the South Tanami all the way down into APY lands. We also did some work out in Mardu country, some water training out there. So we have a big area, again, 600 kilometers from the north to the south. We were lucky with the monitoring of water because we didn't have to reinvent the wheel. We had a methodology already developed for Central Australia. We looked at macroinvertebrates and did, again, a lot of training. And let's not fool anyone. I think I said this earlier. We were trained as much as we trained the local people because this is their country, and they knew the nuances of how to do this work. And we provided some training as well. We had control sites in the national parks that camels couldn't access, and on our asset areas, like this site here in the Petermans. And we had sites in the Simpson Desert after that big rain in 2010. These samples are so different from all our other samples. I'm not going to talk about them today, but I think these were the first aquatic samples ever recorded from the western Simpson Desert. Why use macroinvertebrates to investigate these impacts? Sorry, that should have been a black background. Macroinvertebrates are really sensitive, and if you go to a water hole and do the sampling we do and they're not there, it means something has happened while we were gone that made them disappear. And they're really good because they're used in water watch groups, and most people understand macroinvertebrates, and they're very globally imperiled. And the reason is because we treat fresh water really badly. And I was gonna use a bad word, but I won't. So our findings, in terms of just 2 simple metrics, overall water quality and macroinvertebrates, this is not rocket science. It's like hitting yourself over the head with the water stuff, because what happens when camels are present, the water becomes muddy, animals disappear that live in the water, especially the ones that are sensitive, as opposed to ones sites where camels don't visit. Why would we care about macroinvertebrates? What we find with some of Jenny Davis's work is that at some of these sites, these animals haven't even been described. So we're losing biodiversity because there are endemic species out there that we suspect are there, but they haven't even been described, so they're new to science. Okay. Turbidity. Turbidity is muddiness, okay? So when camels are present, it's a lot more turbid. But what's more nuanced than that, this is a picture of Reg Uluru collecting water from this site that's used quite a bit on a road between Amida and Mutijulu. And on this day, he collected this water to drink, but we did an analysis later, and that water was filled with fecal coliforms. So some of these sites traditionally they used to drink are no longer drinkable, even if they appear clear. Again, species richness drops when camels are present. And why do we care about that? Because this is work Glennis McBurney did in our lab in Alice Springs, looking at camel dung. When camel dung is present, sensitive animals drop out. One of the sensitive animals that drops out are the predators. Mosquito larvae is like honey badger. It don't care, and they just keep going. So without those predators, you even have more mosquito larvae, which in the end could be a health hazard as well if those water holes are communities. How much water are camels drinking? This is data loggers we put in a small water hole down near Mutijulu. These big dips are only from camels. So our biggest native mammals are out there are Euros and red kangaroos. And then when it's wet, dry, they'll drink 100 milliliters of water a day. A camel can drink up to 200 liters a day. That's 2,000 times more. So with natives, you don't get a drop, and with camels, you get big drops. I never thought I would see this happen. The largest water hole we work in is near Docker River. It's a swimming hole for the community of Docker River. In October, we did measurements. It held 980,000 liters of water. If you account for substantial pan evaporation and camels visiting that site, by February it was dry. So that's a loss of 800,000 liters of water from the environment. It would only take 45 camels visiting a day to dry up that water hole. And was that an isolated incident? No. People have been alluding all day to this mass camel mortality that happened at water holes. We know it happened from the South Tanami down into the APY lands, which is a huge area. This is a small spring in the Petermans, worked in there in October. These are some native animals using it. The camels stampeded into that water hole. We know because we had cameras set up in January. And this is the end result. There's Kathy, there's the spring. It becomes ludicrous to make graphs like this because on that day, there is nothing with gills, predators, or sensitive species. It's just over the top, some of the damage they can do. And they don't go alone. These camels will take these native animals with them. We found a lot of dead native animals on that trip. What happens to the native wildlife? When camels are not there, we get more animals visiting water holes. That's not surprising. Camels prefer to be at water holes at night. When it gets dry, they'll go in the day and night. Dingoes like to go at night, but when camels are there, dingoes switch to the day. They visit during the day. If dingoes are there during the day, other wildlife doesn't visit. And these analyses are ongoing. We have a million images from our camera traps. And finally, I'd like to talk about the importance of traditional management. Traditional management increased longevity and surface area, and it had a huge ecological outcome when people were on country managing like they still do. So here's Rini Kulajar and some other ladies from Mutijulu cleaning out this water hole, which is very hard work. So by 4 o'clock, by 4 o'clock, Rini had gotten this cleaned out and it was flowing again. By the next evening, the native birds were using it, including bronze-winged pigeons, and Burke parrots sneak in with bronze-winged pigeons, and we didn't know that before we started this study. 8:30, the camels arrived. They stay there till 7:30 the next morning and basically undo all the work Rini had done the day before. Okay, so the juxta of this is that these things that are impacted here in red, can we push them back up into this healthy state in green? Can these sites recover that have been so badly impacted? Our data suggests here, when you go from red to blue? Yes, they can. So our data so far show that when camels, the pressures ease, they can recover. And that's really significant because in the literature, it suggests that in inland Australia, when sites are really badly impacted, they pass a tipping point and the impacts can be irreversible. We haven't seen that yet, so that's good. So again, what did we have at the beginning? I don't care what the density is really. It could be 0 camels to 1 camel. At these densities, sometimes at a small spring, 1 is too many, and you get these legacy effects. So pushing it down, again, this is the future management. Where are we going to be on this continuum? And I'd like to end with the legacy slide, not just of impacts, but the idea that these people that come behind us, my own daughters as well have done this work with me, the legacy of them having a chance to be good land managers, I think is also important. And I'd like to really make a sincere thanks to all the ranger groups I've worked with that couldn't be with me here today. And thank you very much.